that particular chapter. One major question, if you study, you can answer all the 10 questions.
Okay. Okay.
That is how.
Yeah.
Kindly mute yourself. Anybody else to join? Shall we start?
Okay, ma'am.
Yeah, then fine. So good afternoon. And today's lecture, it is a slightly, continuation of yesterday's lecture, and as well as we have missed 1.1, that is the introduction to cytogenetics. That has not been done. And after that only we should have gone for chromosome structure. As I didn't know what topics have been covered for you, so I started with chromosomes. So today we'll just go for the first one, that is the introduction to cytogenetics. What exactly is cytogenetics? And under cytogenetics only, we will be coming to know about the structure, the function, the inheritance characteristics of chromosomes, then DNA, genes, DNA replication, that is molecular genetics, then cell cycle, chromosomes, nucleic acid, all this, and the diseases associated with the genes and the chromosome, all this come under cytogenetics. It is nothing but selfie of cell. Cytogenetics, it is nothing but selfie of a cell. That means when we are taking a selfie, what are we doing? We are capturing the detailed structure of our face or detailed structure of our dress or whatever it is, isn't it? The same way we are capturing the detailed structure and function of a cell. That is called as cytogenetics. So we will be discussing about the importance of cytogenetics and how is it related to all the other chapters of uh, the syllabus, what you have it. Then we will jump on to the chromosomal abnormalities or chromosomal aberrations. Fine. I'll start sharing. Is it visible?
Yes, ma'am.
Yeah. Clarity. Clarity is there. Okay, fine. Now coming to cyto. Cyto means cell, the genetics of cell. That is the internal structure or the function of the cell. How this cell is playing an important role in the inheritance of a complete structure. So that is what we are going to study. So it is a branch of genetics. It is a branch of genetics that deals with the study of inheritance in relation to the structure and function of chromosome and in their role in human disease and heredity. So this is the definition of cytogenetics. The genetics is a part where we are talking about the chromosome or the nuclear material or the genetic material, how the genetic material plays its important role in the discourse of inheritance or the heredity and maintaining or perpetuating the life. So, how the cell is playing an important role, what exactly the cell consists of, where it is going to lead into the inheritance pattern. So, that is what we are going to study in this. Now, this cytogenetics uh, is related to many other branches of the science. When we are talking about cytogenetics, it involves the study of nucleic acid, DNA, RNA, its structure, its function, how it folds off, and what happens if the DNA is less or more or denatured or it has been transcribed. All these we will be able to study along with the study of nucleic acid. Other one is chromosomes and genes. The cytogenetic study deals with chromosomes and genes also. Chromosome. If we know the chromosome, the number of the chromosome, we will be able to identify the species, what it is. We can have an interrelationship with different species. That is what we have done with karyotyping. We can also do pedigree analysis. Then the cytogenetics also deals with the cell cycle. What happens during the cell cycle? What are the the changes that are taking place in terms of genes, in terms of chromosomes, how the cell is dividing, how the cell is increasing, and when the cell is dividing, when the cell is in interface or a resting state. All that, it is nothing but the gameplay of the chromosomes that will play and thereby bring about the cytokinesis. So cytogenetics also studies of the cell cycle. Then cytogenetics also deals with the cytogenetic technique. These techniques are very, very important to know about the chromosomal abnormalities and the diseases. So when we are studying about the chromosome, we also should know about the abnormalities of the chromosomes. What are these abnormalities of the chromosomes or the diseases associated with the chromosome? That is the chromosome that is there in case if there is any damage to the chromosome that is occurring. How does this damage occur? This damage can occur due to number of environmental factors, chemical factors, physical damage, or the chemical damage, etc. When it happens to the chromosomes, it leads into the deformities of the body, deformities of the functioning. So thereby, to identify this, we need to have the cytogenetic technique. And most important cytogenetic technique is the fish, that in situ hybridization, fluorescent in situ hybridization, which is called as FISH, FISH. It is not there in the purview of your syllabus, but along with this, I will be providing you the steps of doing in situ hybridization, which will be helpful for you to take up any further examination. So in situ hybridization is the latest technology, what we can use it. Apart from that, we have plotting techniques, we have the banding techniques. All these are to study the chromosomal nature, the deformities that are there. So we have to be well acquainted with the cytogenetic technique. Apart from that, when we are talking about chromosome, the most important phenomena or the basis of genetics of or basis of life is the genetics. That is how the chromosomes are being inherited. That is the Mendelian pattern of inheritance. And when we are talking of Mendelian inheritance, I think Mendel's laws, we have studied about independent assortment, law of dominance, etc., etc. All these are the laws and how the deviation occurs in this particular law. So for that deviation, we have also studied about the hybrid ratio, monohybrid ratio, the deviations of monohybrid ratio, that is codominant, then incomplete dominant. And in case of the dihybrid ratio, that modified dihybrid ratios, they are like duplicate genes, supplementary genes, complementary genes. All these are nothing but the inheritance pattern wherein you are seeing that the chromosomes, when they are bringing about the patching up or when the interaction is taking place, you are seeing that the dominant alleles or codominant alleles, they are being expressed in different capacities. When they are being expressed in different capacities, you are able to see different phenotypic conditions and also thereby leading into different genotypic ratios. So all this is included in the study of cytogenetics. So in total, when we want to talk about what is cytogenetics, if a question is being asked, it is the branch of genetics which deals with the structure and inheritance in relation to the structure and function of the chromosome. Its role in human diseases and heredity. It also plays or it has an important links with the study of nucleic acid, chromosomes, gene, cell cycle, cytogenetic techniques, Mendelian inheritance, and chromosomal abnormalities. All this together come under cytogenetics. In order to know the study of the cytogenetics, we need to know certain terminology. Already we are well acquainted with the terminology, but still we have to know about the terminology as and when we are doing some genetics and all that, we need to know these terms. First is the character. What do you mean by the character? Character is nothing but that is the inheritable feature that varies among individuals. Like from each individual, it will be varying it. What are they? Like, for example, color of the eye, color of the hair, height of that person, skin color. All these are called as a character. So what all the phenotypic structures you are seeing in a person, they all come as a character. Then trait. What is a trait? It pertains to each variant of the character. Each variant of the character. When we say color of the eye, color can be brown, blue, green, black, anything. So color of the eye, when we talk about one character, if it is having different the traits, that means it has a different range, then each one is called as a trait. Like for example, height of the plant. Height of the plant is a character. When we say tall or short or tall or dark, this becomes a trait of a character. Then true breeding. True breeding is the parents would produce and they produce the offspring that would carry the same type of the phenotype. That is like begets like, we say. So in that way, that when we bring about the cross, the same type of a offspring will be produced. Hybridization is nothing but mating or crossing over of the two variants to bring about a different phenotypic and genotypic combination. So yeah, Mendel has done the breeding techniques with the pea plant and to study the variability of the chromosomes and variability of the traits that are there, how the genes play an important role in bringing about the inheritance of the character. So there he has done the hybridization of plants, taken two tall plants, two short plants, then first generation tall plant and the parent plant, parent tall plant, first generation with a dog plant, all these he has done. That is nothing but it is a part of hybridization. So in the Mendelian inheritance, we always refer P, F1, F2. These are nothing but P is parental generation, where it is nothing but the two parents have been taken, the pure breed of the parents have been taken for the crossing over to produce the offspring. And the first offspring is called as F1 generation. And then the first one generation, if we take the F1 generation, cross it with any of the parent combination, the second generation, what we get, that or hybrids of the first one generation when we are crossing, the offspring what we get, they are called as the F2 generation. So F1 hybrid or F1 plant cross either with the parent or among the hybrids itself in the F1 into F1 generation, then the offspring what we get, those offsprings are called as F2 generation. If we are doing it with a parent, it can be a test cross or a back cross, that we are not going into the genetics of it. Coming to genotype. Genotype is nothing but it is a genetic composition of a particular individual. Genetic composition in the sense that what kind of genes does it have? Is it having 3:1? 3:1 is, now we are taking only the tall plant and the short plant. But what type of tall do they have? Is it the pure tall or it is a hybrid tall? So when you want to know the segregation of these genes, then those genetic compositions is called as a genotype. Phenotype, as I told you, tall plants and dwarf plants, just by looking at the external feature, we'll say that they are phenotypically same or phenotypically different. So it is the information that is being given by the visibility of the characters. It can be a tall character, short character, color combination, whatever it is, that is whatever we are able to see, those characters are called as a phenotype. Coming to locus. Locus is a point, a specific fixed position on a chromosome where a particular gene is located. Say for tall plant or something, maybe it is the fifth position, or we can say it as a 10th position for height of the plant. So that is a fixed point on a chromosome where we will be able to see a particular character, so or particular trait. Then that point is called as a locus. Allele is an alternative form of a gene. Allele, that is, we have homologous chromosomes. Two chromosomes will be there. If we have a trait for tallness on this particular part, and the same locus, same point will be present on the homologous chromosome. This chromosome, this gene that is present on a homologous chromosome, it is considered to be as an allele. That is nothing but the similar type of a gene occupying the same position. It is the alternative form of a gene. Homozygous, an organism having a pair of identical alleles at a given locus, they are called as a homologous chromosomes. Heterozygous chromosomes. Heterozygous chromosomes or heterozygous condition is that condition if they have different alleles at a given point. Like for example, when we take a heterozygous tall plant, I think all of you remember your intermediate, capital T, small t, small t. When we say capital T, that is a trait for tallness of a plant. and it is present on a homologous chromosome at a fixed position. In case if it is one of the chromosome has capital T and one of the chromosome has the other chromosome has small t, the same location, it is a heterozygous condition. So an organism having two different alleles at a given locus, then it is called as a heterozygous condition. What are genes? The genes, they are the nucleotide sequence that are coding for a polypeptide, coding a polypeptide sequence, which makes an enzyme or a structural protein. That small unit is called as a gene. Gene is nothing but a nucleotide sequence. AUG or AUG, TUG, UAG, all these combination will be forming a set of genes. And this is genes have been represented by a genetic code. We know that how it is being given, that is a three codon code will be there. And these are nothing but they are coding for an amino acid. The sequence of an amino acid will be considered as a gene. So it is a nucleotide sequence coding a polypeptide sequence. This is called as a gene. Now, when we talk about this terminology, apart from that, we should little bit go into the history of it, how the cytogenetic study has evolved. So initially, when you say it was a Hippocratic school, where they say that the organism would assemble into an individual. That means the gametes will take up to assemble everything to form an individual. Like that that is in 1800. The the concept was going on from 1600 to 1700. You will see that the concept was going on in the same phase. So few of the examples or few of the discoveries I have just mentioned here, I'll just read out. Nicholas Hartsocker, he said that the sperm, head of the sperm will contain a miniature human being, which is called as homunculus. Then William Harvey, he has said the theory of epigenetics. According to him, he says that the genes, there are certain factors which will be taking part in the inheritance of a particular character or a trait. Then Robert Hooke, he has discovered the structure of a cell under the microscope. He has seen the corpuscle, which is made up of small unit, which is called as a cell. So he is known to give the structure of a particular cell. Anton van Leeuwenhoek, he is the one who started using the microscope, wherein it has changed or revolutionized the entire molecular study structure. Jan Purkinje, he has described the nucleus within the cell. Robert Brown coined the name nucleus. Then Virchow, he has given the cell theory concept. Gregor Mendel, the hybridization and the Mendel theory. Then Hartwig, he said the fusion of male and female gametes into the formation of a zygote, which will develop into an individual. Then Fleming, he said he has synthesized one particular, then he was a physicist. He has synthesized a dye which will be able to color the chromosomes so that we will be able to view the chromosomes. Waldeyer, he has termed the point chromosome. From 1900 to 1944, there was development that was occurring in chromosome theory and sex determination, etc. From 1944 till present, we are all working on the structure, the function, then denaturation or delocalization or manipulation of DNA, RNA, which is called as a molecular genetics. We are in this particular phase of molecular genetics where we are trying to analyze each and every significant data that is present on the gene to overcome the deformities or the disease studies, etc. So that the conditions which are detrimental to their life can be eliminated out. So here, how the process is occurring and what can be done, that is to remove such type of deformities or diseases from the body, all that is being done in the present generation. That is from 1944 onwards till now. We are developing globally to fight against this aspect, and this phenomena is called as the molecular genetics. At a molecular level, we are targeting the molecule. So our desirable consent so that it will not have the same type of the genetic composition where the population will be totally suffering with it. Now, coming to the cytogenetic part, we can broadly classify into classical genetics, molecular genetics, evolutionary genetics. So, in case of the classical genetics, it is the Mendel's inheritance theory that was a breakthrough for study of the chromosomes or study of the cell, etc. So, it was a breakthrough. And then it led into the chromosomal theory. According to chromosomal, how the chromosomes are being formed, what is the function of a chromosome, how the chromosomes are being inherited, and what is the function, how much of protein it is contributing, how much of food it is generating, how much of ATP, all the functions associated with the chromosome. Now, is it useful for energy process? What is the physiology of it? All this, the physiological process as well as the genetic composition and the genetic aspects, functions of chromosome has been put forth, and the chromosome that is called as, I've summarized it as chromosomal theory. And then during this study, in the classical day, there was a concept of one gene, one polypeptide for the synthesis of an enzyme. So one gene, one polypeptide was the breakthrough again in the biochemical, like biochemical genetics, so that we are able to identify like what proteins can be produced from a single set of the codons or the mRNA sequence that is there. So classical genetics, what we are studying, that is Mendel's law, sex linkage, sex determination, etc., etc. All this will lead into the analysis of pedigree as well as karyotyping. If we don't know the structure of a chromosome, if we don't know like one gene is able to code on for one particular protein, if we don't know how the inheritance pattern is there, will not be able to do the karyotyping. Karyotyping is so it is a phenomena where we are identifying the chromosomes based on from by arranging them in a sequence order, and then identifying whether do we have any kind of a genetic disorder or not, to find out if there is any extra chromosome or if there is a lack of chromosome. All that we are able to get through it. So by doing karyotyping, we'll be able to understand. And the other one is pedigree analysis. Pedigree analysis is that phenomena wherein based on the genetic composition, what we have it, we'll try to trace our ancestors. From where did we come? So that is called as pedigree analysis. What is the lineage of our body? Like from where did we arrive? Who is our father? Who is grandfather? Grandfather's father, father, grandfather's father's father's father. To identify that, we'll have a gene sequence thing that is called as the pedigree analysis, which will tell us the similarities between two things, and then we'll say that yes, we belong to this particular clan or this particular person or this particular family, etc. So that is about the pedigree analysis. Now coming to the next one, that is a molecular genetics. When we talk about molecular genetics, the central dogma of life is the nothing but it is the DNA which is having the capacity to replicate, and that DNA will be transcribed by mRNA, and then it is being transported to protein. So this is a central dogma of life. If protein is not being synthesized, that means the DNA is not replicating properly, nor transcription is occurring properly. Therefore, there is no protein formation, and protein is required for a lot of energy in the body. So the central dogma of molecular genetics is that the DNA replicates, and once the DNA is replicating, it is leading into the transcription through mRNAs and then converting it into a protein. So DNA, the genetic material that is there, this the genetic material, DNA, it is a double helical structure. We all know that Watson and Crick model, which says it's a double helical structure, which is made up of the bases, that is ATGC. Now, DNA, in molecular genetics, we study about the DNA replication, DNA repair, etc. All that will be studied here. We also study about the genetic code, because unless until we study about the genetic code, we will not know what amino acids are there, how the amino acids are being how the amino acids are taking part in the building up of the proteins in the body. Then transcription, translation. All these are certain aspects of molecular genetics where we have to emphasize on this. Then what is the application of molecular genetics? It is having a recombinant DNA technology. It is useful. Molecular genetics, it is providing us with a recombinant DNA technology or PCR, cloning, gene therapy. During gene therapy, we are just analyzing the genes and try to cut the genes or isolate the genes or modify the genes by inserting certain enzymes into it or by inserting certain the agents into it, like that, by bringing about the gene therapy. So uh, The study of this cytogenetics, molecular genetics, leads us to find out the remedy for most of the diseases by gene therapy, by changing the gene, by changing or alternating a particular factor. Say, for example, gene therapy, how this that is, you are targeting only the gene. Suppose I'm preparing a tea. I'm taking two different types of milk. One is directly cow milk, and the other one is I'm taking it from Country Delight, for example. When you prepare a tea, there is a change in the taste from the normal one what we're getting and Country Delight what we are getting it. Now, in case if I take the normal milk and I would like to prepare the same taste of the tea of Country Delight, what will I do? I'll bring about certain changes in the milk that is being taken from a local market. What will I do? I'll try to boil it so much that whatever water content that is there in uh, that milk will be evaporated, and I'll get that thick milk as that of Country Delight, and it may taste the same thing. So the same thing is happening in the gene therapy also. When we know that there is certain defect in a particular gene, or one gene is not functional, we can replace that gene. We can try to change that, translocate that particular gene. So that part of theory is called as a gene therapy, by which we are able to isolate it then and there itself. Then the study of genes again, that is called as a genomic, or what gene therapy, we are in proteomics and genomics. They are omics, they are nothing but the study of genes and the proteins, where we can bring about a lot of drastic changes in this protein expression and the gene performance. Transcriptome, that is when we are studying the enzymes which are bringing about the transcriptases. Suppose it is not functioning, we can modify that, and that study is known as transcriptomics. That is, we are able, omics is nothing but it is a study where we are able to manipulate this particular gene structure. Then the third type of a branch of the cytogenetics is evolutionary genetics. Evolutionary genetics is very, very important because we should know where do we come from and what is going to happen. So it brings about the changes that are occurring in a particular population. Suppose if we say that when we go to North India, we are identified as South Indians immediately because of our style, what we are having. That is in a population, even if we go there, just by the traits or the characters that are there within us, we are able to identify that they are different. Suppose I am going and staying there for more than 10 years or something like that, even then, uh, I will be considered as a local population, but still to identify that I come from some other place, that is the identification that has to that from a population, I need to take out my frequency, how many of them are from South India, how many of them has come from abroad. Like if we want to know that, we should know the frequency. Why we should know the frequency is that if we know the frequency, generally in a population, when we talk about the change in the frequency of the gene, it will lead into mutations. And when there is a possibility of exchange of these na- genetic material, then this may be fixed up in a population. It will be fixed up in a population wherein a new trend or new life will be starting off. So in a population, we would like to know the frequency of any kind of genes that are there, dominant genes, for example, recessive genes that are there. If we know the dominant, that is desirable ones, we can be able to sustain it, and undesirable ones can be eliminated out. So to know that, we should know the frequency, and that frequency study has been given by Hardy-Weinberg equation, which says P square plus 2PQ plus Q square is equal to 1. So here, this equation, or the Hardy-Weinberg equation, give us genotypic frequency. That means in a population, what is the number of individuals which has come from USA, if we take it in a population, if they say that it is 10, that means the primary population is 100. So 100 square plus 2 P into 10 plus 10 square. That will be your formula, and it should come up to 1. That is 50% of this and 50% of that should come, so that it will be a complete set, and we will be able to identify what changes that are taking place within the species, without the species, etc. So the application of evolutionary genetics is that it is bringing about the socio-biological aspects, where we are able to study the behavior under the genetic counter. That means what is the social behavior of an animal, keeping in view the genetic composition of it, and also which is acted upon by the natural selection. We are not artificially allowing the uh, uh, individuals to become mature or to become highlighted or whatever it is. And naturally, it has to be selected. And we will be just studying by using this evolutionary study. It's a time-taking process. We will be able to identify what exactly is happening to the population. So, with the study of Hardy-Weinberg law, we'll be able to find out what are the new mutations that are there. Because when we take the frequency of P gene and Q gene, that is A and B gene, when we are taking, if it is that is recessive and the dominant alleles that are there, based on that, by the ratio what we are getting, if it is more than one or something, there is some kind of a deformity that is occurring, and we can rectify that deformity directly by checking it. So it also leads into the genetic drift. That is the study of the evolutionary genetics lead into the genetic drift. Now, coming to the next aspect. So, till here, what did we study about cytogenetics? Cytogenetics is the branch of science which will be dealing with the chromosomal structure, function, inheritance, and its attributes that are there for the continuation of life, which is involved, or with the study of cytogenetics involved the nucleic acid study, the chromosome and gene studies, then the cell cycle, the importance of cell cycle, the Mendelian inheritance, the chromosomal abnormalities, and also it talks about most of the techniques. When we are talking about these techniques or the cytogenetics, when we go into the details of it, we know that there's a lot of history work has been done. A lot of work has been done in the research of these cytogenetic plants or the material, so that the 1 minute. 1 minute. So that we can be able to fix the population with the required characteristics. So cytogenetics play an important role in the society to identify the recessive and the dominant genes and favorable genes which are taken up by the natural selection. Then coming to the next aspects of it. Is it clear till this part?
Excuse me, ma'am.
Clear, ma'am.
Okay. Now
Excuse
coming,
me,
yes, ma'am.
but we have, ma'am, cytogenetic culture setup and harvest it is there, ma'am. But in this, where it is there, ma'am, genes details, it is not there in textbook.
See, that is, I think
Culture
that is
setup and harvest.
1.7 or something is there. Culture and harvest.
unit 7, it is there, ma'am.
This is
Cytogenetic
introduction.
culture setup and
All
harvest.
that will come in the techniques what we are doing, why we are doing this, cytogenetic techniques. How we are doing that, you are culturing the tissue in a viral substance, or you're taking the blood cells and then culturing it then. Why you are doing all these things? To see certain inheritance traits that are there, isn't it? Any cell structure, what you are doing it, why you are doing, to study the chromosomal structure, to study what is the DNA content that it is having. Is there the manipulation can be done in that particular DNA? That now, if it is a disease, say for example, if you are taking cancer cell, we know that cancer cell, there is abnormal growth of the cancers of the cell. What do you mean by abnormal growth? Now, suppose if there is a cut on your finger and you leave it like that, maybe another 2 or 3 days that cut will be vanished, isn't it? Why? Because the epithelial cells capacity to grow, and once it comes to the point, next point, it knows that it is a place of inhibition, that is called as contact inhibition. Once it contacted, it will be inhibited, and then therefore it will be a normal one. But whereas in case of tumorous growth, what will happen? The cell will lose its identity, that once it has come to the contact inhibition point, it will not stop growing. So once it starts growing, what will happen to the cells that are there? It has been lined like this. Now there is no space for it. So automatically it starts jumping onto these cells and then form 2 or 3 layers of it. Isn't it? So that is, so how it is happening? Because the chromosome, the nucleus is dividing, the cell cycle is continuously going on. Why the cell cycle is going on? Because the accumulation is taking, the nucleoplasmic accumulation, that is the cytoplasmic accumulation is going on. So once it starts going on, so where it has to be stopped? So which gene is active for this? If we target that particular gene, we'll stop the cancerous growth, isn't it? So for that, you are doing all the cultures.
Okay.
Is your doubt clear?
Okay, ma'am.
Yeah. Now coming to the next chapter, that is, I think 1.3 it is. Unit 1, it is 3, that is chromosomal abnormalities. Chromosomal abnormalities, what can we expect in a chromosome? We say that there are 46 number. That is, we are saying set of chromosomes are there. So number of chromosomes. Any change in the number of a chromosome will lead into a different kind of a disease. And that disease, it can be morphological disease or physiological disease. Physiological in the sense that it may interfere with the enzyme activity. Like for example, if we take diabetes, the genes that are responsible for the production of that particular hormone which controls the sugar level in the body is deformed. So therefore, it will not be able to produce that particular enzyme or that particular hormone so that the sugar levels can be reduced in the body. So what is happening if we target that particular gene and replace that particular gene, then automatically we don't require any kind of insulins or any other things to control diabetes. So that is how. So that is physiologically, it is a metabolic blocks what we are seeing. But in case of chromosomal number change or if there is a structural change and all that, we'll see there are a lot of deformities. Why that lot of deformities will be there? Say, for example, I'll be just saying, before going into the chapter, I'll be just saying, so this is my chromosome. Now, in this chromosome, this is for the height of this particular line that is there. It is the trait for the height of my height. And this is for the color. And this is for the intelligence. Okay, IQ level, my color and the height. Now, during mitosis and meiosis, what will happen due to certain changes? Maybe I have taken a lot of radiations or I have been exposed to radiations or some chemicals or something. Maybe due to certain environmental factors. What will happen? This particular position where it is representing for the color of my body is totally lost. That means it can be changed. The sequence, if the sequence of that particular gene is being changed, it will be a different appearance, isn't it? Neither I'll be wheatish, nor I'll be fair, nor I'll be dark. I'll be having some other kind of color. So if there is a change in that particular gene sequence, then automatically it will be there. Sometimes the color for this particular, that is the trait for the height is lost because due to certain thing, this chromosome part has been lost completely. So for me, there is no height. I'll be just like a lump because my body cannot grow. It will grow in the fashion of a lump, like that. So any gene that is there, if there is any change in the chromosome, the genes that are there, the alignment of the gene, if the genes are being aligned improperly, that also it will change. Or if there is a removal of this particular thing, it will be different. Or if there is a duplication of the same type of a gene. For this, I will give you one example. Like for example, you take red bead, yellow bead, green bead. Beads ante mutyalu. Yellow, red, green, and white. 4 beads you take. Yellow, red, green, white. Yellow, red, green, white. Yellow, red. This will be one sequence. If I take 2 yellow, 2 red, 2 green, that will give a different structure to the beads, isn't it? Or if I take 2 yellow, 1 white, 2 green, 2 blue, like that. Each time when we have a sequence of genes that are there, the alignment should be perfect. If the alignment is perfect, then only it will be able to produce a required protein. If the alignment is changed, then it will give a mismatched protein which is not required by our body, and then it will lead into different type of metabolic blocks, and these metabolic blocks will lead into changes in number of structures which we call it as a syndrome. What do you mean by a syndrome? Syndrome is, it is not one single thing that is being changed. Suppose if my diabetic gene is not properly functioning, I will have diabetes. If there is Alzheimer's disease, that is my nervous system, that there is some kind of gene that is responsible for remembering things is lost, I may get Alzheimer's disease. That is only one thing. But when there is a change in the chromosome number or in the chromosome's structure, it will lead into number of aspects. And there's all those symptoms together, we call it as a syndrome. It is affecting many parts of the body, like it will change the height, the shape of the body, the space change will be there. Then you will see that development of different type of organs will be there, or there will be diminishing of organs. All this will lead into a syndrome type of phenomena. So when there is a change in the chromosome, that is the reason why we need to know about what are the changes that can occur in a chromosome when we are exposing ourselves to the radiation. That is the reason why they say that don't expose yourself too much of radiations or X-rays, because they have a high Roentgen power which will be penetrating directly the skin layer, the muscle layer, the bone layer, and then entering the cellular region where it will be damaging most of the cells. The same thing if you are exposing again and again and again, what will happen? The tissue will get damaged. That is the reason why even during the pregnancies, they will say that trimester, then after 3 months, then before delivery, you have to do the scanning or radiation. Beyond that, you should not do it frequently. There's only 1 or 2 they will be taking it. That is, if your frequency of radiation increasing, so there are chances of getting deformities for the chromosome, because they will be exposed directly to the radiation, and because of the radiation they can be cut in the chromosome, or they can be changed in the chromosome, they can go and join to something else. Like that, they can be several type of changes that can happen in the chromosome. So in order to avoid all these things, we should have a knowledge about what changes can occur in a chromosome, which will lead into what type of syndrome. Okay. Now, variation in a chromosome number. Chromosomal abnormalities is nothing but change in the chromosome number or change in the chromosome structure. These two together will lead into the various aspects. So here we should know that what type of changes that will occur. Now, we know that in somatic cells, we have a complete duplicate set, that is 46 chromosomes are there for us, or complete set of chromosomes will be there. And every cell will be a diploid cell because they are getting the genome from both parents, and therefore it is a diploid characteristic, or a diploid set is present. So generally in somatic cells it is a diploid one, but in gametic cells it is the haploid chromosomes or haploid set is present. Now, any variations that are occurring in the chromosome number, it is called as ploidy. Any variation that is occurring in the chromosome number, it is called as a ploidy. If there are changes that are occurring in the structure, we know that there will be deletion or insertion, translocations or duplication can occur. But whereas if there is a change in the number of the chromosome, then that phenomena is called as ploidy. So variation in the number can be of 2 types, euploidy, and the other one is aneuploidy. In euploidy, it is the effect to the entire set of the chromosome. Whereas aneuploidy, it is only one particular chromosome. That is, variation will be occurring in the single chromosome that is there. So we'll just go into the details of it. So what happens if there are chromosomal abnormalities in the body? As we said, the diploid and the haploid chromosome, and we know that there are autosomes and sex chromosomes. They can be changed in the autosomes or they can be changed in the sex chromosome. If there is a change in the sex chromosomes, what type of syndromes will be seeing? If there is a change in the autosomes, what kind of syndromes will be able to see? Now, these are chromosomal abnormalities. Why we should be so much interested? Because it is a leading cause of mental retardation and miscarriages. So therefore, we have to be very careful with the chromosomal abnormalities. Therefore, frequent scanning has to be done. So frequent scanning in a sense that to know before the child is born and all that, they do the prenatal testing, where if there is a case history of any kind of abnormalities, a mental retardation or something like that, if that kind of abnormalities are there, then we can go in for this prenatal testing. So in the prenatal testing, we'll be able to know about any kind of the deformities are there, so we can rectify then and there. So this chromosomal abnormalities, they are the leading cause of mental retardation and miscarriage, and 50% of recovered first trimester abortion, that is whatever trimester means first 3 months abortions that are taking place, these are due to the chromosomal aberrations only. Otherwise, only 1/3 of the conceptions will give rise to live births. Otherwise, the rest of them, they will be going as a miscarriage, or there will be a deformity in the conceiving. 1 in 150 live births, 1 in 150 live births, they are diagnosable with a chromosomal abnormality. That is the frequency. Among 150, we can find one person with a chromosomal abnormality. And those abnormalities we can find out by looking at the phenotypic structure: broad head, mental retardation, no coordination, or no development of the organs properly, or different type of these structures. Based on that, we will be able to identify the changes in the chromosome. So chromosomal aberrations in terms of number of chromosomes can be divided into euploidy and aneuploidy. I said euploidy. If the euploid means the cells have a multiple of the normal number of chromosomes. What do you mean by multiple of the normal? That is, n is the number of chromosomes, 2n, 3n, 4n, 5n, like that. You'll have multiples of the normal set of the chromosome. One set, you call it as a haploid, where you will see it in the sex cells or in the reproductive cell. Then diploid cells, they are present in the somatic cell. Both of them, haploid and diploid, both of them come under the form or the category of euploidy. I'll say it as, it come under the category of euploidy. When euploid cell has more than diploid number, it is called as a polyploid cell. It can be triploidy or tetraploidy, etc. Triploidy is 46 with another set of chromosome 24. So coming up to how much? 46 plus another 24. It should be 68. Triploidy. I have written the 69. Okay. So like that, tetraploidy. 4 copies of each will be there. So 92 chromosomes. Like that, there will be multitude of the chromosomes that will be there. Combined triploidy and tetraploidy, it will lead into 10% of the miscarriages. And then neither triploid nor tetraploid fetuses can survive. Tetraploid, because why they cannot survive? We know that 46 is the limiting number. And if the fetus survives with 68 chromosomes, that means fetus cell will be having 68 chromosomes. And when the reproductive cells are being formed, the 68, it has to become half. How much will it be? 68 half means 30, 34, isn't it? 69 pairs. Are you getting me?
Yes, ma'am.
Yes, ma'am.
When I'm saying it is 36, 34, nobody is responding.
17.
Not 17. 46 is our chromosome. Plus one more set of chromosomes is there. Isn't it? So when one more set of chromosome is there, that is 3n. Triploidy is 3n. So when we have one more set of chromosome, that when it becomes, when sex cells are being formed, 46 chromosomes will be only 23 chromosomes, isn't it? That is 23 in a sense, 22 plus 1. But here, when we are talking about the triploid number of cells, when we take the half, each cell will be having half. That is more the number of chromosomes than 24. It will be having 10 chromosomes extra. So therefore, when it combines with a normal male, you can just imagine what will be the combination of the cell. So 46 plus this 10 will be there. This 10 will not have any kind of other thing. Now, in the next generation, if we say that the person will be having 46 plus this 10, that is 56 chromosomes will be there. When it goes to the next generation, and again, that is not possible because a set of chromosomes is responsible, or it is specific for a particular species. So such type of fetuses, generally, they will not be surviving. The nature will try to eliminate them by itself. Then, coming to one more part of euploidy, I will be talking about. Monoploid. Monoploidy, it is also called as haploidy, where the individuals are generally sterile. Polyploidy, where you will have 2 to 3 sets, that is more sets will be attached to the normal set, that is n, 2n, 3n, 4n, like that. You will be having 3 sets or more than 3 sets. They are called as polyploidy. Under polyploidy, autopolyploidy and allopolyploidy. Autopolyploidy is based on the chromosomes that are there, that is autosomes that are there, triploidy, tetraploidy. Allopolyploidy, it is generally referred to the sex chromosomes, whatever we have it. So in general, we are saying, suppose if you are taking one female with this many number of chromosomes, and then we combine with another type of male, or when the fusion occurs, you will get the polyploidy as 18C plus 18C, which is not advisable. So this kind of true polyploidy is not possible. Generally, it is possible only in plants, but not in human beings. Then coming to the next one, aneuploidy. When you talk about aneuploidy, it is incomplete genome wherein you have either less or more number of chromosomes. A somatic cell that does not contain a multiple of 23 chromosomes, it will not have multiple, but only one or two chromosomes will be more, or it can be less. Like for example, it can be 45 chromosome or it can be 47 chromosome. That is 2n minus 1 or 2n plus 1 chromosome can be seen. The cell containing 3 copies of one chromosome is called as trisomy. Three copies is trisomy. 45 plus 1 plus 1, like that. Or, sorry. Now, 3 copies of one chromosome. Like for example, we are taking 19th chromosome. 19th chromosome, we should have homologous pair, only 2. But we'll be seeing a third one also. So that is how. So in case of the chromosome 21, we will see that they are having, instead of homologous, there will be one more extra chromosome. So 3 copies of one chromosome, that is called as trisomy. Monosomy, it is the presence of only one copy of any chromosome. Generally, monosomy is often considered to be as lethal because there will be only one, there is no homologous pair. So this infant can survive for some time, but they cannot be, like able to survive for a longer period of time, and they will be lethal because the chromosome is totally not there. Therefore, it becomes quite lethal for these particular individuals. 1 minute. They become generally lethal. So this trisomy person, they can combine, they can survive for some time, but later on it becomes quite difficult. So it is better, the survival can be better with more number of chromosomes rather than less number of chromosomes. When there is a less number of one particular set of genes has been totally lost. One set of representation is totally lost. Suppose in that case, what happens? Recessive allele. Generally, recessive allele will not be expressed in the generation, isn't it? Now, when we talk about tall plant and a dwarf plant will be expressed only when it is single or it is having a pair of recessive genes, isn't it? So there you will, or it has been masked, that is the dominant gene has been masked, epistatic gene, then only it will be able to express. So such type of genes where you see that monosomy, only one copy of that particular will be there, the other partner is not there, and that particular condition, when there is a total loss of one particular gene or there is a complete loss of one chromosome, it will lead into lethal genes or epistatic genes, wherein the recessive characters will be expressed, and generally the recessive characters are more lethal and dangerous than the hybrid characters. So like for example, baldness. This baldness can be expressed only when the recessive gene for the baldness is expressed. Unless and until the dominant character is there, non-bald dominant character is there, baldness will not be there. If the dominant character is not there, it has been removed, then the recessive allele is been there, then you will be able to see that. So that expression of the recessive allele will make it lethal. So therefore, we have to be very careful with the monosomic type of syndromes. This is a small example of this. This is a normal cell where each cell will replicate. This will replicate. This will replicate. 4 chromatids will be formed. 1, 2, 3, 4 chromatids are being formed. And then from this is replicating and then forming 2 chromosomes. From here, you will be seeing 1 more chromosome extra. And whereas here, this chromosome should have come here. So because of the non-disjunction of the meiotic division, you're seeing that one cell is with the extra chromosome, the other cell is without the chromosome. So therefore, this phenomena is called as trisomy and this phenomena is called as monosomy. So the individual who is with one extra chromosome will be 23, that is 46 plus 1 chromosome, whereas here it is 46 minus 1. So like that, during this, if there is a non-disjunction occurring where the meiosis is taking place, the cell division is non-disjuncted, then you will see that disparity in the chromosomes will be seen, which is being given to the offspring cells. So we know that this phenomena will bring about the change in the chromosome number. There are other things that like double monosomic. Monosomic, what we said, it is 2n minus 1. Double monosomic is 2n minus 1 minus 1. So two chromosomes will be less. So nullisomic. Now here two chromosomes will be less in the sense that 2n, 46 chromosomes what we have it. In that, the two set of chromosomes will be removed. Maybe one chromosome from one particular cell, that is maybe 19 chromosome is lost and one more will be 10 chromosome will be lost. Like that, there are two different individual chromosomes will be lost. So that is 2n minus 1 and 1. Nullisomic. I think this I don't know. Nullisomic. 1 minute. I think I'll how to read. Okay, I'm not able to do it. Nullisomic means here 2n minus 2. That means one particular chromosome is totally lost. Like AB is a gene sequence. Here ABC, AB. Uh, 1 minute. I don't know how to move this. Further. In this AB. Now here you see that ABC. In this is the gene sequence that we have it. In that C and C is totally lost. We are getting only AB and AB. So two chromosomes are totally lost. Two homologous chromosomes are being lost. There were such type of like change of aneuploidy is known as nullisomic. Polysomic. When we talk about polysomic, here the duplication of one or more gene can occur. So here here we were talking about removal of chromosome, loss of one chromosome. Whereas in polysomic, there can be addition of certain chromosome. Like in case, if there is addition of one chromosome, then you call it as trisomy. So here it is ABC. ABC. This is homologous 2 chromosome. Addition of one more chromosome A. One more chromosome A. That is 2n. ABC is 2n. 23 plus one more chromosome A will be added. Next, double trisomic. In double trisomic, 2n plus 1 plus 1. This is 2n is ABC plus A, and there will be one more chromosome, that is B. Not A will be added. A and B will be there. That is two different chromosomes are being extra added to it. Maybe chromosome 10 and chromosome 21. Now coming to tetrasomic, 2n plus 2. That means ABC is the normal genomic composition. That is double set what you have it. Plus two extra chromosomes of same one. A and A will be the extra chromosome that is being added. Pentasomic, that is 2n plus 3. Same type of the chromosome will be added thrice. ABC is the general composition plus A will be there. Because of this, I am not able to see. I don't know how to bring it down. I'll try, wait. I'm not able to bring it down. Anyways, when I circulate the notes, you will be able to see. The other type of polyploidies are autosomal aneuploidy. When you talk about autosomal aneuploidy, this aneuploidy, we said that it is trisomy. Generally, we are talking about trisomy or monosomy. So, in case partially trisomic condition, we will see there are two conditions that are possible. One is partial trisomy, the other one is chromosomal mosaic. In partial trisomy, what happens? Only an extra portion of the chromosome is present in each cell. Like, for example, trisomy 21, Down syndrome. Similarly, in chromosomal mosaic, you will see that trisomies occurs only in some of the cells, but not in all the cells. Now, in case of partial trisomy, it will be there that all the cells, somatic cells of similar type will be having or the extra chromosome, but whereas in case of mosaic, all the cells will not have the extra chromosome, only few of the cells will be having the extra chromosome. You understand? Yes?
Yes, ma'am. Yes, ma'am.
Okay.
Yes,
Now,
ma'am.
coming to the next, how these chromosomal aberrations occur, that is how the change in the number will be occurring. This is because of the disjunction, normal separation of the uh, chromosomes during the cell division occurs. But in case of these abnormalities, you will see that this normal separation will not occur, and this will cause the aneuploidy. So here, the homologous chromosomes that are there or the sister chromatids, they will not separate properly during mitosis or meiosis. As a result, you will see that when there is the duplication, that is, it is undergoing meiotic division, the kinetochore should break properly, but here it is not breaking properly. As a result, you will see a pair of extra chromosomes. So this is called as non-disjunction. Generally, it should have equal amount of chromosomes to be given to the two daughter cells, but it is not happening. So this kind of phenomena is called non-disjunction. And because of non-disjunction during the mitosis and meiosis, you see the condition called as aneuploidy. Now coming to each one of it, trisomy, Down syndrome. When you talk about Down syndrome, it is an best example for aneuploidy trisomy 21, where 1 in 800 live births, you will find a person with trisomic condition. The IQ level will be between 20 to 70. They have low nasal bridge, epicanthal folds, that is the double folds at the chin regions, etc., protruding tongue, short stature, poor muscle tone. These are all the conditions what you see in trisomy 21. And generally, this kind of risk occurs when the maternal age is more than 35. That is the reason why they say that late marriages and late conception are leading into most of the mentally retarded conditions. Most of the deformities or abnormalities were occurring because now of the hormonal changes that are occurring at the same time, the growth and the physiological metabolism is also changing beyond 35. That is the reason why they say that the maternal age should not be more than 35 before conception. Then aneuploidy in the sex chromosome. This was in the autosome. Now let us see aneuploidy in the sex chromosome. One of the most common trisomy is trisomy X. That means the female will have the X chromosome, and it is called as meta female. They are called as meta females, and this you can see it in Turner syndrome. The symptoms are variable. They will be sterile individuals. Then they will have menstrual irregularities or mental retardation can be seen, and these symptoms can be worsened if there is an further addition of the X chromosome. So what will be the structure or what is the syndrome, what you see, Turner syndrome, characteristics of Turner syndrome? Females are with only one X chromosome. What are the characteristics? Absence of ovaries. You will be seeing that there is a sterility, short stature, webbing of the neck, edema can be seen, underdeveloped breast with nipples, high number of aborted fetuses can be expressed. X is usually inherited from mother. So as these abnormalities, it will lead into the sexual deformities. Therefore, now the change in the X chromosome leads into, and this type of syndrome is called as the Turner syndrome. One more is the Klinefelter syndrome. In this, individuals with at least two X's and one Y chromosome. So therefore, because of the Y chromosome, they are specifically having the male appearance. So characteristics male appearance, they develop female-like breast, that is called as gynecomastia, small testes, spaced body hairs, that is they lack pubic hair, long limbs will be there, testicular in atrophy, infertility will be prevailing, and hip is female-like. Some individuals can be XXY or triple XY. The abnormalities increase with each X chromosome that is being added. Many men with XXY do not know that they are having this particular condition unless and until the maturity time or the maturity level has been reached. Then, so euploidy and aneuploidy, this is change in the number of the chromosomes. If there is a change, deletion of the chromosome number, you will have a different type of conditions. If there is an addition, you have a different type of condition. When there is a deletion, the survival becomes little difficult. That is in the monosomic conditions, etc. The survival becomes difficult because a complete loss of the chromosomal content will be there. Whereas with the addition, they are still surviving with different type of deformity in their physical appearances. Now, coming to the second type of the chromosomal aberrations, that is variation occurring in the structure of chromosomes. Variation occurring in the structure of chromosomes. What are they? There are two types of structural changes we can see. That is, generally they bring about the chromosomal mutations. Why mutations? Because there will be change in the sequence of a particular gene. So change in the sequence of the gene, or they can be at one particular single point, there will be loss of a gene or the impairment of a gene or something like that. So the change in the structure of any part of the chromosome will lead into the mutation. And this can occur because of the change in the physical and chemical change that has been brought about with the physical and chemical agents. Now, this change, structural changes can be taken into two categories. One is changes in the number of genes, which include deletion and duplication. That is removal and duplication, one. And the other one is there won't be any deletion or duplication, but there will be rearrangement of the gene. It can be inversion or translocation. The gene sequence can be inverted, or they can be transferred from one homologous chromosome to another homologous chromosome, or it can be to another heterologous chromosomal structure. So the variation in the structure of the chromosomes or the structural abnormalities of the chromosomes can be divided into two categories. One is the first category, where there can be deletion or duplication of the chromosomes, of the genes, or the chromosome part can be seen. Whereas the second one is there will be change in the arrangement of the genes. It can be inverted or it can be translocated. Let us see what happens in case of deletion or deficiency. During deletion, you see that it results from the loss or part of a chromosome. It is a loss of or a part of a chromosome. The loss may be affecting the end of the chromosome as a terminal deletion, or it can be from the middle of the chromosomal body. So terminal part can be eliminated, or it can be from the middle of the body. Suppose this is a chromosome which is having genes A, B, C, D, and E. And here this D part is being removed. It is being deleted. So you will see A, B, that is, it is being removed. You will get a final product A, B, C, D. Therefore, this gene has been, due to certain changes, due to chemical action or due to any changes, irradiation, etc., you will see that there is a deletion of this particular gene, and the final gene, what you will, with the chromosome, what you will be getting is A, B, C, and E. There's a total loss of a gene, that is D. So it is from the middle of the body of the chromosome. Therefore, that type of deletion is called as intercalary deletion. If it is from the anterior part, then it is called as a terminal deletion. The terminal deletion is produced by a single break at the end of the chromosome, giving rise to small eccentric fragment. Small fragment, eccentric fragment will be there in the terminal one. Whereas interstitial deficiency, it is produced by two breaks. That is below the eccentric region, you will be able to see a small fragment, as well as there will be the lost fragment that is there. And that small fragment that is being removed off will not have a kinetochore, therefore it will be lost in the cytoplasm. Deletion can be detected during the pairing of homologous chromosome. How do you know that the chromosome gene has been deleted? When the meiotic division is occurring, when pairing is occurring. During that, what happens? Homologous chromosomes will pair. Now, what is happening? This chromosome does not have D. The homologous chromosome is having D gene. So, at that particular point, it will form a loop. When there is a looping occurring during the meiotic division or during pairing, you will come to know that this particular gene is not there. Therefore, there are looping or bulging is taking place in the homologous chromosome. And this may lead into genetic defects. And what are those genetic defects? Genes present in that section are absent. So therefore, it will lead into physiological changes that are being done. So therefore, the organism will not be able to survive. So because certain genes are totally being removed, certain physiological actives, suppose if the enzyme which is producing Like for example, for digestion, if the enzyme which is digesting protein is totally lost in the body, the gene which is producing the enzyme for the protein digestion is lost in the body. So proteins cannot be digested in our body. So protein gets accumulated in the body, isn't it? So how much protein can be accumulated in my body? To a certain extent, more amount of protein, I will be having proteinuria. Then protein related diseases, etc. Protein metabolic disorders will be there. And because of that disorders, I may not able to survive. So therefore, if these deletions are bringing about any physiological changes or if they are responsible for any physiological activity, then that organism or the animal cannot survive. And moreover, due to deletion, suppose this D is a dominant character, that is the dominant character is there and the recessive character is present over here. If the dominant character is being removed, this recessive character will be expressed. When this recessive character is being expressed, it becomes lethal. So that kind of recessive character expression, it is called as a pseudo-dominance. Example for this, parent with a recessive character is ABC, ABC. With a dominant character is ABC and ABC. Now, to this parent has been irradiated, exposed to certain radiation, X-rays. So what happens here? The gametes that are being formed, both the gametes will be ABC for the recessive one. But whereas for this, one gamete will be ABC, the other gamete will be AC. So when there is a fusion, you will see ABC and small ABC. So here in this individual, you will see the dominant character is expressed. So this individual will be normal. Whereas due to irradiation, one of the gene that is been lost here, if that gamete is reacting with the recessive one, you will see that B portion that is being lost. This is recessive. The B is been lost from this. So that phenotype will be expressed as a recessive one. It might be lethal or it can be dangerous or it can be anything. Therefore, such type of occurrence or expression can occur due to deletion of the chromosome. And not only that, when you talk about deletion, this deletion will bring about the breakage or loss of the DNA, and it is similar to that of Cytogenetics syndrome. Deletion of short arm of the chromosome of 5. Deletion of the chromosome 5, short arm of chromosome 5 lead into Cytogenetics syndrome. And it will have the same type of symptoms. Like, for example, low birth weight will be there. The second one is mental retardation, microcephaly. That is, brain will be a little smaller, microcephaly. And then microcephaly, and then your growth retardation can be seen. So due to deletion, you will have the similar type of syndromes like that of the Cytogenetics syndrome. Coming to duplication. Now, this duplication, it is nothing but the presence of a repeated gene sequence. That means the same gene sequence will be repeatedly present. Therefore, such type of conditions are called as the duplication. When a section of a chromosome is present twice, it is called as duplication. And then duplication in the same reason as that of Cytogenetics causes mental retardation, but there won't be any physical abnormalities. Here, example we can see here. This is one chromosome. This is the homologous chromosome. A, B, C, D, E, F, G. These are the genes. Here also A, B, C, D, E, F, G are the genes. When the crossing is occurring, during this crossing, the F gene that is there, it has gone to the second chromosome. See, the crossing that has occurred, you have seen that F has gone to this particular chromosome. Now, when there is a break occurring at the centromere, you will see that when the break has occurred, the FG has come to this. And then there will be sticky ends of this end. Therefore, A, B, C, D, E, F is lost. G. Where is it lost? It is being given to the homologous chromosome. So, this gene is duplicated. F gene is duplicated. So, when there is a synapsis occurring, when there is a the pairing occurs, at that time this F will form a loop which shows that this gene has been duplicated and there is no F gene in the homologous chromosome. Is it clear? So in duplication, you will see that there is an increase in the number of the gene sequence, or duplication of the genes will be taking place and the number can be twice or thrice repeated. Then tandem duplication, the different types of duplications are there. Tandem duplications, you see, A, B, C, D, E. We are seeing the duplication at this particular point. That's why I put a dot here. F, G, H, I. These are the gene sequence. Now, duplication is occurring for DE. DE is duplicated. Tandem, that is automatically somewhere some parts have been done. So this is called as a tandem. The same gene will be repeated again and again. So that is called as a tandem duplication. Reverse tandem duplication. In reverse tandem duplication, you will see that DE is there. Reverse tandem. So ED, the duplication will be ED like that. This duplication will be taking place. So in my gene, now this gene is coding for one protein. Now with a change in these two, it will definitely change. Like for example, I'll give you one example. We are preparing a lemon juice. In a lemon juice, what we'll be taking? We'll take water, sugar, and then we will take lemon. This is lemon juice. But in that, if I add a pinch of salt to it, completely changes the taste of it, isn't it? Similarly, by addition of one or two genes, that totally the protein expression will be changed because the ATGC banding that is occurring here, there will be a mismatch of the banding that is occurring. So therefore, it will not be able to produce a functional protein. Then coming to the next one, displaced duplication. Displaced, it is being displaced from one position to another position. Say again, EF, we are talking about the duplication, that is displacement that is occurring in this particular. So this is a centromere and this is a long arm, this is a short arm. So in the same arm, suppose the duplication is occurring here. A, D, E, B, C. B, C, D, E. D, E is after A. Duplication has occurred here. So instead of A, B, C, D, E, we are getting A, D, E, B, C, D, E and FGL. So this is called as homobrachial duplication. It is occurring in the same arm. Suppose if this DE comes here in the short arm, ABCD is long arm, normal, FGHDEI. When it is having the duplication in the other arm, it is called as a heterobrachial displacement. And there is another type of displacement which is called as transposition. It will bring about the transformation, this duplication in another chromosome, not the homologous chromosome, but another chromosome. Then that is called as transposition. Duplication on non-homologous chromosome. For a non, this is one chromosome A, B, C, D, E, F, G, H. Other chromosome will have genes KLMN. So here, if this DE goes into the other chromosome, then it is called as a transposition type of duplication. Generally, we see this kind of duplication in polytene chromosome. There is one more effect which is called as a position effect. Position effect is one of the important aspect of the duplication where we are able to see that the changes, the addition of one particular gene into it brings about a lot of changes. What are they? One classical example is Drosophila. The gene for eye shape and development is located on 16A chromosome. 16A of X chromosome. Normal eye is recessive with 780 facets and it is round in shape. Wild type, it will have two bar regions. Heterozygous bar region, and this I will explain it to you in terms of this. Now you see these are two homologous chromosomes and this is a local location for the eye color. This is normal. In case of a bar body or in case of a wild type, you will see that there are another position. One more position is there for the bar body. So it should be below this region, but one more extra is there. This is a bar. This is a wild type. Now, when there is a duplication occurring, this duplication, it is occurring on a homologous chromosome. Here you are getting double bar. Instead of one bar, you are getting double bar. So here, in this case, you are able to see a duplication. And when you see the eye structure, you will be able to see like this. This is a normal one. This is with a bar. Just to see this eye structure, how it is changing. When it is a double bar, the eye structure is totally changed. So there is a change in the shape of the eye with the change in the locus of the genes of a eye color in Drosophila. Is it clear? So it is occurring at one particular, this is brought about due to difference in the position of the gene. Difference in the position of the gene. Therefore, it is called as a position effect. So it will bring about the difference in the phenotypic ratio. So therefore, it is called as a position effect. So what is a position effect? That is, it brings about the change in the difference in the position of one particular gene. It is called as a position effect. Example is Drosophila, eye color or eye shape. wherein which is present on the X chromosome at 16A locus. And yes, yes, another 5 minutes. I will stop. Next one is translocation. As I told you, deletion and duplication is one. Translocation is, here it is decrease in the number of genes or addition of the number of genes. But here it is only transporting from one position to another. Transfer of the segment. It is a transfer of the segment of chromosome to a different part of the same or different chromosome. Here we can say it can be between homologous chromosome or non-homologous chromosome. It can be at a simple terminal segment or it can bring about the complete shift. Let us see that. So this is the normal chromosome here, pair 1 and pair 2. If there is a shift, translocation, you can see this uncolored state and here it is a color. So a part of this chromosome has been shifted to this. So it is a homologous translocation that has occurred. From here to here it has translocated. Whereas in this case, it is not to the homologous chromosome. It is towards the hetero. This is chromosome 1, chromosome 2, chromosome 3 and chromosome 4. This should go to chromosome 4, but there is a translocation occurring between 2 and 3. So this is not a homologous chromosome. So it is called as a heterozygote or translocation occurring between two different chromosomes. So translocation, it can occur between homologous or non-homologous chromosome. The next one here in this, you can see the shift translocation. This is one chromosome with A, B, C, D and E. Now, when you see this A, B, C, D, if due to certain reasons, there can be a change or shift in the chromosome. So you can see here, A instead of B, it is C, D, B and E. This B has been shifted to this part, A, C, D and B, E. This is totally been shifted within the chromosome. The gene position has been shifted within the chromosome. Whereas in this case, there are two different chromosomes, A, B, C, D, E and F, G, H, I. These are two different chromosomes. This B gene in this is been shifted to the second chromosome, F, G, H, I. This B is being shifted to another chromosome, non-homologous chromosome. So they can be the reciprocal chromosome. That means it is being shifted to non-homologous, or it can be simple terminal end can be changed to the non-homologous chromosome, or they can be shift of the genes onto the same chromosome or with a different chromosome. So you can see here, if you can find out, you'll be able to see that the gene B has been added to the second chromosome. And generally, this you will be seeing this in case of the chronic myeloid leukemia, where there will be a loss of chromosome 22, which is also called as Philadelphia chromosome. Because of this kind of shifts that are there, the complete chromosome will be lost. That is, it becomes non-functional because of the change. Here I'm showing only 1 point, but generally the genes will be 2 or 3 genes will be totally gone. So that particular chromosome becomes completely nullified, or it loses its function. The last one, what we are talking about is inversion. It is the inversion. In inversion, the gene sequence will be inverted. It occurs when a segment of a chromosome becomes changed by rotation of 180 degrees. And as a result, the complete sequence is in a reverse order without changing any number of the genes. A, B, C, D, E. A, B, C, D has changed. A, B, C, D. So now when it is going for a looping, you will be see that the synapses, the loop will be totally different and there will be 2 breaks occurring in this particular condition. So there will be a complete change in the reverse order. And this also will bring about the position effect. You can see here A, B, C, D, E, F, G, H, A, B, C, F, E, D. There is a completely reverse order of this particular gene is being done. So here inversion has occurred between D and F. And this particular type of inversion will lead into the racial differences. And also it may lead into overlapping or bringing about changes in the overlapping and crossing over of the genes, where you will find different type of gene sequences occurring. So it interferes with the overlapping of genes and crossing over genes during the cell cycle or meiotic cycle. And this is the leading cause for the racial differences in the chromosome. Last point I would like to say is fragile sites. The chromosomes, because of the change in the structure, you will see that they are prone to be broken. That is, the breaks that are there, they will be frequently breaking. So areas on a chromosome that develop breaks, that particular area is called as a fragile site. And this you can see it during the culture of the cell. When you are culturing the cells, you will see that the chromosomes are breaking, and that site is called as the fragile sites. And generally, it will not have any kind of a relationship with the disease, but it will bring about different type of syndromes. And that syndrome is called as a fragile X syndrome. It is more dangerous than the Down syndrome because it will lead into mental retardation. Is it so? Yes, ma'am? Yes? Somebody was telling something. This is the end. Yes? Total, how many students are there today?
16 members.
16 members. Take the screenshot and send. I will be sending this PPT also. I'll arrange it in proper order and then send it. Is it okay? So what did we, I'll just summarize what we studied today. What is the significance of cytogenetics study? Where exactly the cytogenetic study is responsible and why we need to know about the cytogenetic study, because we can know about the chromosomal aberrations or changes that are occurring, to know about the syndromes, the hereditary patterns, etc. Then coming to the chromosomal abnormalities. What are the chromosomal abnormalities? They can be in either in the number of the chromosomes or it can be in the structure of the chromosome. Number of the chromosomal abnormalities, it is called as a ploidy. You have euploidy, aneuploidy. And in case of the structural changes, you have deletion, duplication, translocation, and inversion. Fine? Yes?
Okay, ma'am. Okay.
Any doubts anywhere?
No, ma'am. No, ma'am.
Is it clear?
Clear, ma'am.
Yes?
Yes, ma'am.
Yes. Okay. Anybody, any problem with the language problem or something, you can just tell me.
No, ma'am.
No, ma'am.
Okay, it's clear.
Clear.
Okay,
Yeah.
fine. I will be more comfortable with the handwritten notes rather than this printed notes. Printed, all words appear to be same, isn't it?
Yes,
So,
ma'am.
yeah. So I'll just, if you're comfortable with this, I will go with it. I'll try to bring about more colored pictures and all that. Otherwise, we'll go for a PPT. Fine. Is it okay?
Okay, ma'am.
Okay, then shall we leave? Call it a day today?
Okay.
Come
Okay,
along.
ma'am. Thank you, ma'am.
Yes. Any doubt? Somebody was asking something. Anyways, if you're feeling free to ask, please ask. Okay. So, good evening. Thank you for attending. Please send me
Good
the
evening,
screenshot.
ma'am. Thank you, ma'am.
Good evening.
Thank
Okay,
you,
ma'am.
ma'am. Thank you, ma'am. Thank you, ma'am.
Thank you.
Hello, ma'am.
Hello.
And did you send it to Kishore sir, ma'am? All the notes of tomorrow,
No, I told you.
yesterday?
No, I told you. No. Before next class, I will be sending it.
Okay, ma'am.
Next,
Because
because
there are so many classes, ma'am. 4 class a day, and there is nothing, any notes have provided to us, ma'am.
okay. No,
No any
because
notes.
I have just written it upside down. No, it may be little confusing for you. So I'll just prepare it in a proper format and then send it. If you are okay with the
Okay,
same thing,
ma'am.
I'll
Okay,
put it
ma'am.
in the same thing. Okay.
Ma'am,
Fine. Yeah.
very nice. The lecture was very nice, ma'am. Very good.
Thank you.
Thank you, ma'am.
Okay.
Okay, thank you.